Hi, good morning. My name is Tori Guarino, and this summer I work towards optimizing an E. coli expression system for Penium, margaritaceum, Rubisco. Can the lights dim? Or... Thanks. Okay, so my summer revolved around Rubisco, which is the most abundant enzyme on Earth. So its job is to fixate carbon dioxide from the atmosphere through the process of photosynthesis for plants. So just because it's the most abundant doesn't necessarily mean it's the most efficient. So Rubisco's issues with efficiency stem from the fact that it can catalyze a competing reaction in the atmosphere with oxygen gas, which is over 500 times abundant as carbon dioxide within the atmosphere. So Rubisco's low turnover rate and poor specificity to carbon dioxide cause it to be a major limiting factor in plant growth. So to study Rubisco this summer, we used Penium. So one of the reasons we chose this as our model organism is because it has a pyrenoid, which most green algae have, but land plants do not have, with the exception of hornworts. So a pyrenoid is very important for photosynthesis because it is a carbon dioxide concentrating mechanism. So basically, carbon dioxide is pumped into the pyrenoid and increases carbon dioxide concentrations around Rubisco, which combats the battle between oxygen gas and carbon dioxide that I talked about earlier. So pyrenoid's ability to increase the efficiency of photosynthesis caused many scientists to be motivated to work towards engineering pyrenoids into land plants, such as crops. So another reason for choosing Penium as our model organism is that it's an immediate sister to land plants. So Chlamydomonas, another algal species, has been a popular model organism for pyrenoid and Rubisco studying, but its divergence of over 1 billion years ago has hindered its efficiency to be engineered into land plants. So as an alternative, Penium serves as a more ideal candidate due to its more recent divergence to land plants and its status as a sister species to land plants denoted by the star. And some land plants I'll talk about during the talk are Anthoceras agrestis, which is a hornwort species, Arabidopsis, and then tomatoes, which is a crop plant that would benefit from pyrenoid studying. So Penium as an algal culture takes about 8 weeks to grow to a point where the Rubisco and its pyrenoid can be studied and transformed and modified. So as an alternative, we will be using an E. coli expression system to produce Rubisco. And an E. coli expression system has been published and refined using Arabidopsis. So during the summer, we will be modifying that Arabidopsis system to pioneer it with Penium in order to produce Rubisco. So in order to study pyrenoids, we first need an effective way to produce Rubisco. So this summer, using the E. coli expression system, I will be trying to optimize production of Rubisco. So this is how Rubisco is made. It's a very complicated process with many parts and pieces, but they are all necessary in order to produce a properly formed Rubisco. So using 3 plasmids, transforming them into E. coli so that E. coli expresses the genes and produces all these parts and pieces, we can rapidly modify and purify Rubisco. So the first plasmid encodes for the large and small subunit, which come together to form the LAS8 molecule, because Rubisco is formed of 8 large and 8 small subunits. And there's also a hist tag on the small subunit of this plasmid, which is a series of histidines, which will be used for protein purification later on in the methods. The next plasmid encodes for the chaperonins, CPN for short, and this helps with folding of the large subunit towards the beginning of the assembly line. And then the PCDF plasmids encodes for the assembly factors and the chaperones, which help with further assembly of the large subunits together. So in order to match the Arabidopsis system, there were certain criteria for the Penium design plasmids to meet in order to be candidates. So the sequence length and percent identity to the Arabidopsis system were important, as well as the target peptide and expression of the Penium genes. And this Penium plasmid system was established by my mentor, Bryce, prior to my arrival. So my role was to use it, to analyze it, and to optimize it. So in totality, the system works as such. So these 3 plasmids are integrated into the E. coli, and then the Rubisco is grown with all the parts and pieces, and then it's harvested from the E. coli. So with the proteins made by the E. coli, now purification is the next step. So in order to purify the Rubiscos from everything else, the His tags are really important. So the His tags attached to the small subunit have an affinity to nickel. So when we run it through the IMAC, the immobilized metal affinity chromatography, the nickel is able to keep the proteins from eluting. So the affinity allows for everything else to be eluted besides the things with the His tags, which are the Rubiscos, right? So then after everything else is eluted, these proteins are eluted into fractions. And then these fractions are then ran on a gel. And I'll be talking about two different gels today, and I'll explain them when I go over them. But I'll be talking about SDS and native gels. So this is an SDS gel, and it will show both the small and large subunit. So here you see the Arabidopsis system, the Fuller's system, compared to the designed Penium system. So first we noticed that they both have small subunits at around 14 kilodaltons for the molecular weight. And in the Arabidopsis system, we see the large subunit, but we don't really see it in the Penium system. What we do see is CPN60B at around 60 kilodaltons. So if we remember, CPN60B is important for folding the large subunit, so we can assume that the large subunit is being misfolded by the current set of chaperonins on the Penium plasmid. So upon a structural modeling, we can see that the Penium CPN60B is a little dodgy, has a few red flags on it, right, compared to the Arabidopsis system. So I don't know how to use the laser, but yeah, all of this, all the perimeter is a little hairy, you know, so we don't want that. So as an alternative, we are going to use the Arabidopsis PET11A from the published system within our combinations. So now we have the two Penium plasmids compared to, or with the Arabidopsis plasmid, right? So what we notice is there's still, and then also in this column, this is purified Rubisco. So this is what we're looking for within our gel, right? So we see that there's small subunit being produced, just like the last one. And then we see a small amount of large subunit, which is great, which is exciting. But we also see a large amount of CPN60B again, right? So but now this tells us that different combinations can yield different results. So now that we know we can use Arabidopsis within the system, now we're going to try Anthoceras agrestis. So Anthoceras agrestis is a hornwort species, which is a land plant with a pyrenoid. And a postdoc in the Gun Lab Zen allowed me to use his plasmids within the system to just try out more combinations. So I tried a total of 6 combinations this summer. So with varying PCDF and PET11A plasmids. And then you can see in combination 6, I edited the Penium PCDF plasmid. And this edit comes from a suspicious insertion seen within the Penium plasmid that's not within the Arabidopsis system. So to combat this, we isolated it and removed it and shortened it to more accurately model the Arabidopsis PCDF RAF11 or RAF1. So with these 6 combinations, we can compare the different amounts of things produced. So this is a different type of gel. So now instead of seeing both the large and small subunit, we just see Rubisco. So you can see that there's not really too much that follows along that line, but we do see other parts and pieces, right? So the dark black bands can represent the chaperonin complexes by their molecular weight. And then we also see a contaminated small subunit lower down denoted by the green. And we also see 2 unknown bands. So something to note is that within combination 3, this unknown is very prominent compared to everything else, right? And this is the combination that used 2 agrestis plasmids. So in order to better identify what this unknown is, more research can go into the agrestis plasmids. And then also with the edited PCDF plasmid, we see total difference in these bands. So just that short sequence of amino acids that was taken out really modified and affected what was being produced right here. So in totality, you see that all 6 different combinations yielded different results in every category. So future directions is determining those impurities. And this can be done with size exclusion chromatography, which will further separate these proteins in order to better identify them, and also a spec assay for enzymatic activity to allow us to know if Rubisco is actually being produced. And then also using the Penium cultures, the actual algal cultures, to grow it up and harvest the Rubisco in order to better study the Rubisco, to know its parts and pieces, to see what's actually expressed in order to better improve the plasmid system for Penium. And here are my acknowledgements. I'd like to acknowledge Bryce and Laura for their help in the lab this summer, and then also Megan and Delaney for making the RU memorable.
Almost hunches about what those unknowns are, or any directions that you want to start taking to find out what they are?
I sat down with my mentor and another postdoc in the lab, and they didn't even have any predictions. We kind of sat there, I think for like an hour, and we were just kind of looking at the gel, and we just kind of were like, you know, we just need to run it again and do different combinations in order to better identify it. So.
Thank you so much.
Hi, thank you very much for your talk. I would actually want to ask a couple questions, or really just one question mostly, about the protein purification process with those unknowns. So you said in the future directions, you do plan on doing size exclusion chromatography to sort of better isolate that unknown, but also to clean up that Rubisco sample. In terms of the latter there with cleaning up the Rubisco, not necessarily identifying the unknown, do you think using a different kind of immobilized metal chromatography might also work? I know you said you use nickel, but I do also know cobalt works with his tagged proteins. And I was curious if you maybe considered trying a purification with cobalt in the future.
Yeah, that could work. I think just the one that we have at Wheel Hall is the nickel one. And then also another path to take is tagging the large subunit as well. I think that's what Quinn did in his project. So doing both like large and small could help further identify what that is too, being eluted.
Thank you.